Hello everyone, welcome to Fundamental of Biochemistry series. This is episode 4 in which we are going to discuss types and importance of the disaccharide. In last episode we discussed structure and property of the monosaccharide. So let's discuss about the disaccharide. Disaccharide are most abundant which are composed of two monosaccharide unit which join together by glycosidic bond. Here you can see this is one monosaccharide, second monosaccharide. Both are joined together by glycosidic bond. By condensation process and release the one water molecules. Right? So let's discuss about the different examples of the disaccharide. Disaccharides are sucrose, lactose, maltose, cellobiose, and trehalose. In this, sucrose is made up of glucose plus fructose, lactose glucose plus galactose, maltose glucose plus glucose, cellobiose and trehalose also made up of glucose plus glucose. But their glycosidic linkage is different. Here you can see that maltose alpha 1,4, cellobiose beta 1,4, and trehalose alpha 1 to alpha 1. Right? In this example, sucrose and trehalose both are non-reducing sugar. As we discuss in monosaccharide, non-reducing sugar, it means there is a no free aldehyde and no free ketone group is present. So these are the both are the non-reducing sugars example. Remaining are the reducing sugar. Let's discuss about the sucrose. Sucrose also called as the common table sugar, cane sugar, beet sugar or invert sugar. It is a non-reducing sugar. It made up of glucose plus fructose. This is the formula of the sucrose. Sucrose is found in all photosynthetic plant like major it present in sugarcane, beet, maple, pineapple, carrot, etc. When we discuss about the physical property, how you have to remember, just visualize there is a sugar. This sugar, when we observe it, it looks like a crystal. So it is a crystal. There is a no color. There is a that's why we called as a colorless. Odorless, when smell it, there is a no odor, so it is a odorless. When we taste it, it looks like a sweet. So sweet in taste. When we add into the water, it's soluble in the water. So these are the property of the sucrose, physical property. When we discuss about the chemical property, when we heat the sucrose beyond 186 degrees Celsius, it decomposite and converted into caramel. This is a brown in color caramel when we heat the sucrose. This process is known as caramelization, and that is used in topping nowadays and on the chocolate or cake, right? This sucrose, when it break down by the sucrase or invertase enzyme, which are present our intestine, and that converted into glucose and fructose in a separate monosaccharide. So when combustion of the sucrose is produced carbon dioxide, carbon and water molecule. This is the structure of the sucrose. You can see that this is Fischer formula in a linear form, and when we dissolve into water, it converted into the cyclic form. So this is the 5 number carbon and this is 1 number. So 5 number carbon hydroxyl group is attack on the carbon number 1, on which OH group, and from the fructose there is a H that is from the fructose, and by the condensation process remove water molecule and it form the glycosidic bond between this 1 and 2, and it will convert into the sucrose. So this is the structure of the sucrose. What is the function? It acts as a sweetening agent. We are adding various kind of food. It is component of the diet. It is an important part of in our diet, and which will, when it break down, which form the glucose that required for energy. Sucrose digests in our stomach by the invertase or sucrase enzyme, which convert glucose and fructose. In plant, sucrose is transport this sucrose in the throughout the plant. So it is a transport into the plant. If excess amount of sucrose it we take, at that time it promote the diabetes. Overconsumption of sucrose, it may cause the tooth decay and cavities. These are some example of artificial sweetener, which are saccharin, aspartame, sucralose, neotame, stevia. These all are the examples of the sucrose, which are faster, more time sweetener as compared to sucrose. So these are the when these are the examples that are used in food industry, as well as those who are diabetic patient, they are use this artificial sweetener. So most artificial sweeteners are bind to sweet taste receptor on the taste bud cell. So all diabetic patient feel that they are eating this sucrose, like a sugar, but real that are no calorie, that does not have a calorie, but sucrose has a calorie. So its function is reduce the calorie, diabetic management, dental health, weight management, and also used in a food industry. So these are the artificial sweetener. Let's discuss about the maltose. Maltose is a malt sugar which made up of two glucose molecules by glycosidic bond. This is the structure of the maltose. The maltose are for maltose from the sprouting barley, which are present in glucose syrup, honey, caramel, snake bar, fruit wheel, and white bread. It is produced during the digestion of the starch. When starch are going to digest by a enzyme amylase, which break down into the maltose. Maltose are two identical sugar which maltose is which further break down into the glucose to glucose molecule into the intestine by using the amylase enzyme. So here you can see that this is starch. Alpha amylase will be break down into maltose. Maltose further break down into intestine by maltase enzyme, and which convert to glucose unit, right? What is the property of the maltose? Maltose are crystalline, solid in form. This is the 160 to 165 degrees Celsius melting point, soluble in water, and it is a dextro rotatory. Reducing sugar, it form osazone when we react with the phenyl hydrogen. This is the structure of the maltose. This is glucose and glucose, beta D glucose and alpha D glucose, both are joined together and which form the maltose structure. This is a function, it provides energy. It also involved in germination and seed metabolism, food industry. It also act as an intermediate in starch and glycogen digestion. Its role in brewing and fermentation process, when starch will break down into maltose by using of the yeast, which are converted into ethanol and carbon dioxide. It is a clinical diagnosis that maltose tolerant tests were historically used to assess pancreatic amylase and intestinal maltase function. Deficiency in maltase can cause carbohydrate malabsorption. So these are the function of the maltose. Let's discuss about the lactose. Lactose, also called as a milk sugar. Lactose is converted by using the lactase enzyme into glucose plus galactose, which is found in the milk of mammals and animal origin. Human milk contains 6.6 to 8% of the lactose. Cow containing the 4.8%. We can say that mother's milk is about 1.5 times sweeter than cow milk, right? It is made up of galactose and glucose. So this is a structure. It is amorphous, colorless, milky odor, less soluble in water and less sweeter than the sucrose. Melting point is 203 degrees Celsius. It is dextrorotary, reducing sugar. It reduces cuprous ion to Cu plus ion and form osazone crystal like a cotton ball shape. This is a structure. Here you can say this is linear structure, Fisher formula, and this is Haber's formula. Here you can see glucose and beta D galactose, which will give the glycosidic bond and which will give the lactose. What is the function? Major sugar in milk, it is lactose, which is present in mammal's milk. It provides the energy. Lactose are hydrolyzed by lactase enzyme. It convert glucose and galactose, which provide the energy for our baby, for our health, for human being as well as for newborn babies. So it also support the beneficial gut microbiota. Undigestible lactose can be fermented by intestinal microorganisms which are present in intestine, which which produce the beneficial short chain of fatty acid. It also help in calcium utilization. Lactose can enhance calcium absorption, particularly during infancy and childhood, supporting normal bone growth and development. It is reducing sugar. Lactose has a free anomeric carbon, so it behave as a reducing sugar and give positive uh, Benedict test, right? So lactose is a natural sugar of the milk. It provides energy, support growth, promote the gut microbiota. It helps in calcium absorption. It is a reducing disaccharide. Let's discuss about Trehalose and Cellobiose bios in a very short. Trehalose, it is alpha 1,1 glycosidic bond is present. Here beta 1,4 glycosidic bond is present. So where it present Trehalose? Bacteria, fungi, yeast, insect, right? And it hydrolysis of cellulose and plant cell wall. Digested via Trehalase enzyme. Here it not digested by human, but it digested by microbial cellulase enzyme. So when cellulose, which breakdown, which converted into cellobiose, this is cellobiose, which further breakdown by microorganism, and which give the energy and carbons involved in carbon cycle. It is a main function is trehalose stress protection. It also like various kind of heat, osmotic stress, desiccation. It involve in structural breakdown and it generate the energy. It use trehalose used in food preservation, freeze drying of vaccine protein, cosmetic, while this cellobiose used in biofuel, bioethanol production, cellulose enzyme in using research. These are the MCQ for important various kind of exam that you have to solve it. Thank you very much for listening this episode. If you like this episode, then subscribe it, like it, and share.